So our next presenter is Vavra. She's going to talk to us about plants and how they compete with one another.
My name is Vavra. I worked with Mario Bretfeld on a project called The Clone Wars, a study of plant competition. You might be wondering what plant competition actually is. It's defined as the interaction between plants for light, water, carbon dioxide, and soil nutrients. When environmental resources become limited, the plants must compete with each other to obtain what they need in order to actually grow and function as they need to. What are clonal plants? They're defined as a group of genetically identical individuals. They originate vegetatively, not sexually, from one single ancestor. The individuals you see above ground are called gametes, but they remain connected underground by a single, what we like to call runner. They're all clones of the same plant, and you can see in this diagram, you can see right here, these are the ramets, but they're all connected along with a single runner, and this is their same root system, so they're all actually the same plant. It just shoots up the ramens above ground, so it looks like this. As opposed to clonal, we have non-clonal plants that reproduce using two fundamental processes. We have meiosis, which rearranges the genes and reduces the number of chromosomes, and fertilization, which restores the chromosome to a complete diploid number. And most plants do this via pollination. Here's just a general diagram of non-clonal plants in general, not specific to mine. But you can see just the general process that goes through here as it develops seeds from the fruit. And the single seeds germinate, and you can see the single root system of the seedling as it turns into the mature plant. But it only has a single root system for the single plant. Why are we doing this experiment? We would like to observe the competitive potential of a clonal and non-clonal plant. This study will help fill in a gap in our knowledge by studying two very closely related plants. One is just a subspecies of the other. There actually is no known study conducted using two such closely related plants. Most of the time it's an aspen tree versus a non-clonal grass. Nothing that's actually a similar plant. Our main goals in this experiment are to help develop natural weed control strategies and to obtain optimal yields for clonal plants such as bamboo or strawberries. Like strawberries, you can see it's a clonal plant, but the runner is above ground as opposed to underground. I'd like to introduce you to our species. Our two main species are Festuca rubra, which is a clonal grass, and Festuca rubra commutata, which is the non-clonal subspecies of that grass. Our main competitors are Tertium arvense, which is a clonal weed, and Volpia octoflora, which is just a non-clonal grass. Festuca rubra is commonly known as creeping red fescue. It reproduces clonally, and it has many uses such as erosion control, ground cover for lawns, golf greens, turf, and wildlife habitat. Here's just a sort of a diagram. You can see here is the runner, and these are all the ramets shooting off. And you can see the flowers off the grass, but you can see where it would connect onto other ramets and continue along. This is just a general picture of what the plant actually looks like. Festuca rubra commutata is commonly known as chewing fescue. It reproduces non-clonally via pollination. It's used for similar purposes as Festuca rubra, simply because it's such a closely related plant, such as erosion control and ground cover. So here's another diagram. You can see it's the single root system of the plant and the single plant itself. And again, you can see the root system of this one single plant. They're just such similar plants. They both have excellent shade and acid tolerance. They're very resistant to turf disease. They're very hardy grass. They can survive with minimal water and fertilizer, and they grow best in cool climates and high elevations, so they really thrive on like the high plains heartland areas. Our first competitor, Tertium arvense, commonly known as Canada thistle. It reproduces clonally, and because of its invasive clonal behavior, it's actually classified as a noxious weed. You can see the bristly leaves, as well as just actually what it looks like. Volpia octoflora is commonly known as 6-week fescue. It reproduces non-clonally via pollination. It's just another common prairie grass. You can see another diagram with the single root system again and the flowering of the grass, as well as the electricity. For our experiment, a total of 60 pots were set up. 34 of these contained the Vavra rubra clonal species. 17 of those were placed in competition with our clonal Cirsium arvense, 17 with the non-clonal Volpia oxiflora. The remaining 26 contained the Vavra rubra commutata, the non-clonal grass. 13 of those were placed in competition with the clonal Cirsium and 13 with the non-clonal Volpia. We placed them each on a single watering table and we randomized them. By randomized, I mean we didn't put numbers 1 through 60 in line because say 1 through 34 is also super clonal. We wanted to separate them so that although we are in a greenhouse, the conditions change. One spot in the greenhouse may have a higher temperature, one part of the watering system may shoot out more water, it might be hotter, receive more sunlight. So we did this just to ensure that in case there were differences, at least one group, one of each setup would receive the same treatment. The watering system was set up to water once a day for 2 minutes. The length of the longest leaf and the number of leaves in each pot were measured with a centimeter ruler and recorded. After 3 days, the Cirsium arvense and Volpia oxiflora were added to the pots. This was done just to ensure that the Vavra and the Vavra clonal and non-clonal could establish themselves within the pot so that we would actually have good samples to use for our experiment. When this new species had established itself, only the largest was left in the pot. We carefully selected the smaller ones and picked them out to ensure that this one strongest individual could really get the best nutrients it possibly could. After 3 weeks of this setup, our measurements were taken again and recorded. This is an image of our setup. You can see here's like number 2 and 7. They're just really randomized. And the watering system is the sprinklers that shot out, kind of watering all different pots. Here is an image. This is our Vavra clonal. Here was Cirsium arvense. This is the Cirsium. And here is the Vavra rubra. We tried to get just a couple of ramets of each Vavra in the pot for the clonal, and then we planted 4 of the competitor in the top pot. And here it is with the Volpia oxiflora. You can see this is the Volpia, it's much smaller. And here's the Vavra rubra. Here is our commutata, the non-clonal, with Cirsium arvense. Here's the Cirsium, and here is the non-clonal Vavra. And here it is with the Volpia. It's a lot harder to tell the difference between these two just because they are so similar. But again, there's 4 planted of the non-clonal Volpia as well as the non-clonal Vavra. For our results, after the 3 weeks were over, we ran 2-tailed t-tests on the data we took. There was statistically significant data found in the tests concerning the Cirsium arvense growth and germination. Unfortunately, that was the only significant data we found. All of the data concerning Vavra growth and Volpia growth and germination were not significant. Here's our first graph describing Vavra growth in centimeters. The average growth in centimeters is on this axis, and this was one of our not statistically significant data with a p-value of 0.81 and 0.23 respectively. This just shows that the Vavra growth, either the clonal or the non-clonal, was not actually affected very greatly by the competitor plant. But it did show that both Vavra species did grow better in comparison to the Cirsium. Here's our average competitor growth in centimeters. Again, how many centimeters along this axis. This is a statistically significant result. For the Cirsium arvense growth, we have a p-value of 0.02. And this was our first exciting data point. Unfortunately, the Volpia was not affected, but the Cirsium was greatly affected by the Vavra. And our average competitor germination, 10%. Here's the percentage along here. This is our second significant data point. It shows just that the Cirsium was negatively affected by the species competition with another p-value of 0.02. Again, the bulky was not affected, but we're starting to see that the specific clonal really does have an effect on the Xerxes marvensis clone. For our main discovery is that the specific rubra has little competitive advantage over the non-clonal version of the specific rubra, but Xerxes marvensis has a lower germination and growth rate when in competition with this clonal specific rubra. So it shows that the clonal graft may actually have competitive advantage over other clonal species, maybe not necessarily non-clonal. Why this might be, clonal species in general utilize their resources and don't always allow the other species in the pot to obtain what they need. And we had planted the Pacifica rubra clonal several months earlier just to ensure it would grow, just to be sure that we had an effective plant for our sample. And because of its size, it also may have just taken all of the nutrients, but it also could have shaded the other plants in the pot and prevented them from getting the sunlight or the heat that they needed. The Pacifica rubra non-clonal, the commutato, was planted only 4 weeks early, therefore it wasn't as developed as the clonal. Some possible errors need to be accounted for as well. I personally selected the Xerxes marvensis seeds by hand from tiny dried samples, and I would guess that the seeds were probably like 2 millimeters long, this tiny, and it was hard to distinguish between what was necessarily a good seed and what was like a half a seed. So it's possible that some of these seeds may have not been any good just because they weren't from a seed manufacturer, and by some weird chance, all of these were placed in the clonal pots, so they were not able to germinate and grow as well. And as much as we tried to randomize the pots, the conditions in the greenhouse just may have varied too much within that small area that they didn't all receive the same treatment. For future endeavors within this experiment, I think given more time, more significant data may arise. You can't really make assumptions based on 3 weeks of plant data because none of the plants are actually at their mature state. So if they were given enough time to reach the mature state, you would be able to see how they actually grow to a full extent and actually use their resources. On a larger scale, with more than the 60 pots we had, you would be able to look past these random occurrences and see more relevant data. We had thought, looking at the graph, you'd think that there were more significant results, but just the statistical error, it varied. We had some way up high and some really low, so that was just an average. When the experiment is actually taken down in September, the plants will be dried, and the biomass of the roots and leaves will be measured to just see where the nutrients are stored in each plant. And after this process, I'm sure different results will become more apparent just because you'll get a way better analysis just than what you get from just measuring. We also took data points about a week after this set, and the germination rate data was completely different. It wasn't significant anymore. So given more time, again, some results may be nullified, some new results may arise. And what this means, our goal was to develop natural weed control strategies, and with our 2 significant results in the Xerxes germination and growth, this may be possible. We've shown that Xerxes marvensis does not grow well with other clonal species, so we possibly, theoretically, could control the Xerxes marvensis as well as other invasive, toxic clonal weeds if you plant a thriving native clonal grass in this thistle-ridden area. It may be able to choke it out just because it's a stronger competitor. And this could provide a means of restoring natural habitats for these natural species if you could eradicate the invasive. I would like to thank the Adolph Hirst Foundation for sponsoring my research as well as my stay here at FSI. I'd like to thank the University of Northern Colorado for letting us come here, the Frontiers of Science Institute, my mentor Mario Bretfeld for all of his help, Lori Ball, Tony Gondra, Crystal Meisel, and of course, I'd like to thank Frank Skepka for always finding a way to get us in the greenhouse even though none of us had access. I think Tony Otum and Chip Skating Rink for that. I really appreciate all the help that everyone's given throughout this process. Tony has read over my paper thousands of times, and it's been there to support me and let me hang out in his lab when my mentor was gone. So I just really appreciate everything that everyone's done to help me. Are there any questions?
Because earthly martensia is not just weed, but it doesn't do well against coastal, why is it still so invasive and so prevalent in society?
Okay, the question why the surfing is still prevalent even just, can you repeat that? Just
Yeah, sure. You mentioned that earthly martensia was not just weed,
okay.
but it is choked out by chloroquine. There's pleasant why is it that earthly martensia is so prevalent in society today if it's not?
I would assume just that maybe it was less, it might not be the specific grass that it's paired with, so this specific grass made it to be a really strong competitor compared to others, and I really would just guess that it's placed with a variety of clonal and non-clonal, so it's really not given the opportunity to be choked out by a specific plant like this. Right?
How did you take that to the plant?
Like the Cercium with the Zika? They're both they're both prairie species, and they're commonly found together, just in certain areas. So it was just to see if actually planted together, if the clonal graft could actually eradicate these clonal thistles. Yeah?
So the study seems that it's basically about generational plants that can grant multiple generations are allowed to produce new seeds and have those seeds germinate with new vectors as well.
Absolutely. I think that would be a great step forward just to really get more significant information, because you really can't tell over one lifetime of a plant and over not even a full lifetime. So I think if you could really take the time to watch it kind of develop, you would get more significant information. Thanks.
My personal experience as an owner of a home, I can tell you that clonal patients do not have regular growth rates. They seem rather like they're exponential, some sort of multiple status. What has your mentor told you about these September planned tests? Does he anticipate that the tests will reveal that his hypothesis was accurate?
Just from these first results, he does actually have a lot of confidence in the fact that we might find something interesting with this clonal, just because of how fast it's already showing that the clonal graft is choking out the clonal thistle. So we are expecting something pretty cool to come out of the September scan. Yes.
So, do you know exactly the clonal technology to go? Will you get two different plants you were able to cover that runner on the underground runner with those? So was the one plant become two plants and they that way too?
Just from gardening experience, I think yes, because you can cut a piece of the plant out of the ground and transplant it to another area, and it will grow and thrive there as well. So that is a really good way to spread these plants, and also a good way to accidentally spread them out to food. Okay, Bill?
So would it be able to not do my job and check out any seeds?
Theoretically, yes. We would actually need to test it further to recommend you go out and do this, but it is a native graft to this area, so it would not be harmful to go out and plant a particular grass in your lawn, and hopefully it would choke out whatever thistles are in the area. Are there any questions? Thank you.